Hello everyone, welcome to our next video. So today we're going to be looking at chapter 2D and 2F. So we've got a little bit out of order, but I think it will hopefully make sense when you see why. And we're looking at gene expression and protein production. So in our last lesson, we went through proteins, what they do, their basic structure, and DNA and what that does in its basic structure and why they're so important to our lives. Okay, so DNA holds the instructions for all the protein our body needs to make and function. But how do we actually go from those DNA instructions that we looked at to a functional protein? Okay, so that's what this session is going to be about. We're going to go through all the steps that happen to create a protein that can be used by the body. This process is happening all the times and is regulated super closely, which is what we're going to be covering in our next lesson. So let's dive into the protein synthesis pathway. So our first dot point for this one is understanding the genetic code. Okay, so we need to be able to understand the language of genetics and the genetic code in order to be able to understand how these things are used to create a protein. So that's what we're going to start with. Once we know how to read a genetic code, we're going to go through transcription and translation of this code to form a functional protein. So before we start, what are genes? Okay, let's just do a quick recap of what a gene is, because that is what's going to be turned into our protein. So a gene is just a genetic code that is stored within our DNA that will code for a protein. Okay, so genes are what codes for proteins. Most genes are similar amongst humans, but some are different, okay, and that's what makes all of our different characteristics, like, the pigment of our eyes, etc. These instructions code for specific amino acids that will join to become proteins. So hopefully you remember from our last lesson, amino acids are the building blocks. They form the primary structure. That's what we're going to be learning how to create today. And the production of the functional gene products as to proteins is called gene expression. Okay, so going from a DNA sequence to a functional protein is called gene expression, and the protein of gene expression. Before we can understand how a genetic code is turned into a protein, we need to be able to read the genetic code, because the genetic instructions, it's like when you're trying to put together IKEA furniture. If you can't read the instructions, you're never going to put together the coffee table or whatever you're trying to make, and it's the same for this. Okay, so we need to understand this language first. So what is the genetic code? We've got three different versions of the genetic code here. Okay, we've got the DNA code, then the mRNA code, and then the amino acid code, and we need to be able to read all three of those. So the first one is the DNA code. So remember the last lesson we looked at DNA. So in a double-stranded DNA molecule, this would be double-stranded, and it would have our complementary base pairings there. But for the sake of this, we're just looking at one particular strand. We're looking at the template strand of DNA, which I'll go through in a second, because that's what we use to make mRNA and then eventually the protein. So you'll see that this DNA is separated into threes, and we call these triplets. So the DNA code is read in triplet form. So we read DNA in threes, and that's really important to note. When a DNA triplet is transcribed into mRNA, these triplets become codons. So remember how I said that DNA is trapped within the nucleus. It can't perform any roles. It's very precious to us. We protect it. So what we do is we transcribe it into something called messenger RNA, which can move around. It's a temporary molecule that is basically not a replica of the DNA sequence, but it's a readable version of the DNA sequence, okay, that we can use to make proteins. So these DNA triplets are transcribed into mRNA codons. Okay, so in a DNA it's triplet, in an mRNA it's codons, and we'll talk about how it's transcribed a little bit later on. So these are codons. Again, they are read in threes. Do note the change, okay, from a T to a U in the RNA space. We then, this mRNA is then read, okay, also called translation. Okay, so think about, what that means in, our language. When we translate something, we are trying to read it to make sense of it. So this mRNA is then translated into our amino acids, which forms our primary structure that we learnt about the other day. So these are the three important steps of our genetic language that we need to understand. We need to understand that DNA is read in triplets. We need to understand this is transcribed into mRNA codons, and these codons are translated into those amino acids and that primary sequence. So this is called the codon table, and don't let it scare you. It's a lot, and you don't need to remember any of this. You'll never have to recite this. If you have to use this, they'll always give you a copy, okay? But it's important to know how to read it. So the codon table can be used to determine which amino acids are associated with which codon. So remember, I said that mRNA is read in codons, okay? How do we know which codons match with which amino acid? This is how, okay? We read a table like this. So the mRNA strand is used to read this, okay? So when you are reading these, the codons here, always be using the mRNA strand. Don't use the DNA strand. Don't use any other strand, just the mRNA strand. So, and then we've got AUG, which is our start codon. So that is going to make a bit more sense when we look at transcription translation. So this is the codon that is always at the start of all gene expression, and it gives us our methionine amino acid. And then we've got these things called stop codons as well, okay? So again, this will make more sense when we go through it, but a stop codon is what terminates gene expression. So start codons start gene expression, stop codons stop gene expression, okay? So this is the codon table. Now let's go through how to read it, okay? And how to transcribe and translate all of these languages we've learnt about. So let's say I have a DNA strand, okay? I'm just going to write a random DNA strand. Actually, no, I'll start it properly. I feel like I should probably start it properly. We're going to start with TAC, GGG, CTC. Okay, I'll just do three triplets for now, just to make it a bit more straightforward. So this is the DNA sequence that we have, okay? So this is what we call the template strand of DNA, because remember, DNA is double-stranded. This is only one strand, so where's the other strand? This is the template strand. There is the other strand, of course, which is called the coding strand, but we don't use that for transcription translation. We use the template strand. So this is the template strand of DNA. Now what we need to do is we need to transcribe this into mRNA codons. Okay, so how do we do that? So we've got our first triplet here, TAC, and then all we need to do is think of what the complementary base pair is going to be for this base pair. So for instance, the complementary base pair for thiamine, hopefully remember for the other day, is adenine. Okay, so that's going to be the complementary base pair there. Our complementary base pair for adenine is uracil, okay? So this is RNA, so it's uracil, not thiamine. And then for C, our complementary base pair is our guanine. Then guanine again, so it will be cytosine, 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 and then guanine, adenine, guanine, okay? So all I've done there is I've just paired up the complementary base pairings that we learned about in our last lesson, okay? So that's how you transcribe the DNA template strand into an mRNA coding strand, all right? Now, it is this coding strand that we're going to use to make the amino acid sequence, okay? This is our primary sequence that we learned about in our last lesson. So our first codon is AUG, and as we know, it should always be AUG because this is our start codon, okay? So the first amino acid most of the time, I hope, is going to be that MET amino acid, okay? But just for example's sake, how did I read that from the codon table? So the way we read this is we read the first base. So the first base is reading along this left-hand side. Our second base is reading from the top, and our third base is reading from the right-hand side. So the first base in this codon was A, okay, so I'm going to be reading here. Our second base was here, so I'm going to be going down where these meet up, so here. And then I'm going to be looking at our third base from this direction, which was G. So I'm going to be following that all the way over to our first amino acid. So I'll show you how I've done that for each of the other ones as well, okay? So our next triplet, or our next codon, sorry, is CCC. Okay, so how I'm going to read this table, I've got C as my first base, I've got C as my second base. So if I follow this along, okay, and down, it's going to take me to this little subsection here. And then my final base is also C. So I'm going to follow that along, okay, and it is going to give me this amino acid, which is pro, okay, or proline. All right, so that's my next amino acid. And then for my last one is, my last codon is GAG, okay? So I'm going to be looking at my first base as G, my second base as A, and my third base as G. And if I follow all of those along, okay, it's going to give me GAG is here, which is going to give me the glue amino acid, okay? And there we have it. That is our primary sequence, okay? So remember the primary sequence that we looked at in our last lesson looked like that. This is just the letter representation of that, okay? So that's how we go from our DNA to our mRNA to our amino acid sequence using this beautiful codon table that you were going to have to use a lot and learn to love, all right? So now that we know how to read genetic language, okay, and transcribe and translate a genetic sequence, let's see how that actually looks like in practice. So what does that actually look like inside of a cell? Okay, so we're going to be going through gene expression step by step, starting with transcription. Okay, so the first step of gene expression is transcription. So the goal of transcription is to produce this mRNA that we've been talking about. Okay, so DNA is precious. We don't want it to leave the cell. So we make this temporary instruction booklet, which is the messenger RNA, that is able to leave the nucleus and is a copy of those instructions so that we can make our protein. Okay, so that's the goal of transcription. So transcription starts with initiation. Okay, so initiation just means the start of something. All right, so to begin transcription, proteins called transcription factors bind to the promoter. So promoter, we're going to learn about it in our next lesson, so don't stress for now, but basically it's just a region of DNA where things can bind to. Okay, so these transcription factors are going to bind to this promoter region, which is going to allow something called RNA polymerase to also bind to this promoter region. So again, we're going to look at RNA polymerase in more detail later. For now, all you need to know is it's an enzyme that helps to make this mRNA molecule. Okay, so that's what RNA polymerase does. This signal, so these transcription factors, along with the RNA polymerase binding, causes a break to the hydrogen bonds between DNA strands. So remember when we were looking at our DNA, and this is a very simplified, I didn't do the helix or anything, remember that our bases are connected with hydrogen bonds, okay? So to break apart our DNA strand, we need to break those hydrogen bonds, which is going to cause something that looks like this picture here. So you'll see that these hydrogen bonds are intact and then they are broken apart, all right, causing these two strands to separate. One of these strands, like we talked about before, is called the template strand. So this template strand is going to be what is actually read to create the mRNA. And then we've got our coding strand. So that's just the other strand. It's called the coding strand because it has the same code as the mRNA that's going to be produced. So this causes the DNA helix to unwind, unzip, and leaves these nucleotide bases exposed. Okay, so you see how they're both exposed, especially the template strand. So it's leaving it exposed to create this new RNA template strand. So that's what happens in initiation, how this process starts. We then have elongation. So elongation just means making something longer, which is what we're doing with our RNA that we're producing. Okay, so RNA polymerase is going to move along the template strand. So this is our template strand here. It's going to move along the template strand and it's going to read the DNA template strand for the instructions, okay, because that's what it's for. Then we're going to have these free-floating nucleotides. All right, so remember we're going to have our G, our A, our G, our U and our C nucleotides are free-floating in the nucleus. They're going to be joined together to make this mRNA strand. So something very important to note at this point in time. This elongation step produces pre-mRNA. So this is not the final product yet, okay? This process produces pre-mRNA, and we'll talk about how we get to that final mRNA step a little bit later on, but it's really important to note this is pre-mRNA for this phase, okay? So we're adding the bases and making this pre-mRNA molecule. And then we have termination, which is just a fancy word for stop, okay? So how do we stop this process from occurring? We reach something called a termination sequence on the DNA. Again, we're going to learn about this in our next lesson. For now, all you need to know is that it terminates this process. Okay, so once this termination sequence is reached, the RNA polymerase is going to release itself, and it's going to release that pre-mRNA, this pre-mRNA that it's made into the, the nucleus or whatever, and then the DNA is going to wind back up again. So the DNA, which is currently, left bare, is going to reform those hydrogen bonds and wind itself back up into its beautiful helix once more. Okay, so that's how transcription finishes. And then we've got this pre-mRNA molecule that's been produced. So how does it go from pre-mRNA to mRNA? So this process of RNA processing needs to happen. Okay, so to go from pre-mRNA to RNA, we need to have RNA processing. So this only occurs in eukaryotes. That's important to note, especially for our next lesson when we go more into prokaryote stuff. So RNA processing only happens in eukaryotes. Okay, so humans, animals, plants, whatever. And it mostly involves this step here. Okay, so we've got our pre-mRNA, all right, that we made during transcription. To make it a fully functional mRNA molecule, we add what we call a methyl G cap. Okay, so you can see that here, a methyl G cap. So it's just a modified guanine little hat that we put on the end to stabilize. And we put a poly A tail on the end. Okay, which is, as the name suggests, it's just a bunch of A's, a bunch of adenine bases tacked onto the end for stability. Okay, because as it the pre-mRNA, it's just got, there's nothing holding it together. Okay, so we put this cap on and this tail on for stability. Okay, to fully enclose our mRNA molecule. All right, so that's what happens in RNA processing. What also happens is the removal of introns and splicing of extrons. So we're going to learn about this in more detail in our next lesson, but for now, okay, you can see in this pre-mRNA molecule up here that we have these purple bits. Okay, these are our exons, and we have these white bits. These are called our introns. Okay, so exons are the bits we want. All right, so they are the bits that code for the protein. The introns are not filler DNA, but they're there for other purposes like stability and etc., but they don't code for a protein. So what we do in RNA processing is we splice the introns out, we remove them, and we splice the exons together so that the final mRNA product is only exons, because they are the bits that code for the protein that we want. Okay, so that's what happens during RNA processing. What can also happen in RNA processing is this concept called alternative splicing. Okay, so while we're removing those introns and exons and all of that sort of stuff, we can do alternative splicing. So sometimes exons can also be removed, or the order can change during the splicing process. So while we're splicing introns out and splicing exons together, we can do this in several different ways. So we might, the cell might completely, it might just be normal. It might include all exons, as with the first example here. So you can see all these exons in the RNA are all joined together. This is all normal. That's fine. What we can also do is we can remove exons as well. So you'll see in this example here that they've removed this pink exon here has been completely removed from the DNA altogether. And you'll see here that the blue or the green one has been completely removed altogether. We can also rearrange the order, okay? So if you've got, exon 1, 2, and 3, these can be rearranged during this alternative splicing process to create a completely new, different mRNA strand. So this means that a single pre-mRNA molecule can produce many different mRNA molecules depending on which exons are spliced out or rearranged. So this is what alternative splicing is. So what this allows for is a single gene to give rise to many mRNA strands. So why would this be useful? So if we have one genetic strand, okay, and it can code for many different mRNAs and many different protein products, that's really efficient, okay? That's really energy efficient. It means we're using less room within our nucleus. So one strand of DNA can code for many different proteins with this alternative splicing event, which I think is pretty cool. So once we have transcribed, okay, so once we have turned our DNA instructions into a messenger RNA that has been processed and spliced, then we go to translation. So the goal of translation is to read this mRNA that's just been produced to create our polypeptide chain, or the primary structure of the protein that we learned about in our last lesson, okay? So translation has the exact same 3 steps that transcription does. So it has initiation, elongation, and termination. But they look a little bit different, obviously, between the processes. So the initiation in translation, so what happens is the mRNA exits the nucleus and travels to a ribosome. So all of this translation happens within a ribosome. So ribosomes are the main site for translation, which is important to note. So ribosomes are an organelle within our cell, hopefully you remember from year 11. So they're the little protein producing units that we find in our cell. So this mRNA is going to bind to this ribosome and is going to be read until that start codon is recognized. So remember when we were looking at our codon table, that AUG start codon, that is what initiates translation. So we're going to read our mRNA until we reach that start codon, which is AUG. So once that's recognized, we can begin translation. So tRNA, so hopefully you remember from the last lesson, tRNA delivers amino acids. So that's its whole job. And tRNA has something called anticodons. I can't spell. There we go. All right. So tRNA molecule with a complementary anticodon will bind to the ribosome as well. So you'll see that the tRNA possesses UAC. Okay. So hopefully you note that this is complementary to our start codon and our mRNA. So this is called an anticodon. So it's what's complementary to the codon, okay? And this is how it reads it and how it brings over the appropriate amino acid. So once the tRNA reads it with its anticodon, it brings over the appropriate amino acid, which is methionine for this one, because that's our first amino acid. That's how translation begins, okay? So that AUG really needs to be recognized. We then have elongation. So in this scenario, elongation, again, it means the the lengthening of a molecule, but the molecule in this case is our polypeptide chain, or our primary structure of our protein. So the mRNA molecule continues to feed through the ribosome. So once it's got that met amino acid, it continues to be fed through the ribosome. And every codon on the mRNA matches with a complementary anticodon on tRNA, and the tRNA will continue to bring over amino acids that are appropriate, and they will be joined to the polypeptide chain, okay? And these are where our peptide bonds are joining each amino acid over and over again to form that primary structure. Remember that primary structure that we looked at in our first lesson that looks like this? These are our amino acids, these are our peptide bonds, okay? So what's happening, you can see in here, so we've got our codons down the bottom, and we've got our anticodons on our tRNA, and you can see our tRNA is bringing over the appropriate amino acid for the codon. So they're just like a delivery service, basically. They'll read it, they'll go away, get the amino acid, come back, join it on, go away, grab another one, etc. Okay, and this is how our primary structure is built. And this continues. So this will continue until termination. Okay, Termination in this case is when we reach a stop codon. So cast your minds back to that codon table that I showed you. All right, remember there was the start codon, but there was also a couple of stop codons as well. So that is how this process terminates. So once a stop codon is read, the polypeptide chain or the primary structure is finished and it's released into the cytosol. Okay, and then it starts its next journey, which we're about to go through, but that is how genes are expressed. So that is how we get from, we've got our DNA here, okay, this unwinds, our pre-mRNA is produced by adding nucleotide bases, so this is our pre-mRNA here. Then we go through RNA processing, where we are adding all of our stability components, and we might alternatively splice, etc., to get our fully fledged mRNA. This then travels over to the ribosome, and the ribosome will thread the mRNA through itself. tRNA will come over, it'll read with its anticodon, and it will bring its amino acids over until eventually the stop codon is reached and our fully formed polypeptide chain is released into the cytosol as a functional protein. Okay, so that is the magic of gene expression. So we've made this protein, right? Sometimes we'll keep it within the cell because the protein might be needed for something inside the cell. That's cool. But a lot of the time, what we want to do with those proteins is we actually want to export them outside of the cell because we know that proteins have different functions, all over the place. They might be mounting an immune response, they might be building a muscle tissue, etc. So most of the time we need to release that protein out into the extracellular environment to do its job, okay? So the last component of this video is going to be looking at how we transport that protein to the extracellular environment to do its job, okay? And it's called the protein secretion pathway. So the goal of this is to fold, modify, and package proteins for export out of the cell to perform their jobs, just like I said, okay? Obviously, the first part that we just went through, and I'm not going to go through it again, we start at the ribosome, of course, all right? So the proteins are synthesized here, and we just learned how this whole thing happens, okay? So this is technically the first step of protein transport. The protein actually has to be made through transcription and translation. Then what happens is the folding, okay? So remember, we learned about this in the last lesson. We had our primary structure, which can then go into our alpha helices or our beta sheets, which then folds into a tertiary, and then maybe, if it needs to, joins into a quaternary structure. All of this folding happens in the endoplasmic reticulum, okay? So think back to your year 11 organelle knowledge, okay? This all happens inside the endoplasmic reticulum. So it normally happens in the rough endoplasmic reticulum because remember the rough endoplasmic reticulum has ribosomes attached to a surface. So if we are making a protein that we know we're going to export out of the cell, that translation process is normally going to happen inside one of these ribosomes that is attached to the rough endoplasmic reticulum because it makes sense because we want to conserve energy, right? So all of this protein folding that we talked about in our first lesson, primary, secondary, tertiary, quaternary, happens inside the endoplasmic reticulum. Okay, so once it's made, it folds there, and then a transport vesicle is going to bud off and it's going to float to its next location. Okay, so once it's all folded, it's going to go to the Golgi apparatus. So we are looking at this part here. So it's going to travel from our rough endoplasmic reticulum to our Golgi apparatus, where it's going to be further modified and packaged. So when I say modified, they might add chemical groups, they might add sugars, they might remove molecules. So proteins, often need some additions to function properly for whatever their function is. So this is where all of those additions and removals are going to happen. In the Golgi apparatus, it's going to modify, okay? So what happens after all this is done is the Golgi apparatus is going to package these ready-to-go proteins into things called secretory vesicles. And these secretory vesicles are going to be involved in exocytosis of the protein. Hopefully you remember what this means. So that means that the vesicles are going to fuse with the plasma membrane and they're going to release the protein from the cell. Okay, so these secretory vesicles, they fuse, the protein gets released to do all of its very good jobs. Okay, so this is a form of bulk transport, which you might remember, and it's also active transport. So it requires energy. So that's why we're trying to conserve energy in all the processes before it, because it's quite a taxing process. So our protein is now made, and it can now be released to do its jobs. So please come to class with any of your questions, and I'll see you there.